So we're going to take a look today at what plants are doing in the light. In order to do that, we've already taken some test tubes and filled them up with water and added BTB, or bromothymol blue. Now, Lizzie, you're familiar with this from your experience with DNA.
Yep.
My students might be familiar with this because in 7th grade they did some work with this.
Ooh.
One of the cool things about BTB is that it changes color when exposed to carbon dioxide. So these tubes are just plain old water, and I'm going to have you take the straw, put it into the tubes, and blow into them. Then we're going to cork it up, and we're going to notice the change in color. Okay? Okay. And that is why we wear safety gear. Who's there? Come on. 3, 2, and 1. Aren't you glad you did it? 3, 2. Okay, so we can see that the BTB has changed color when exposed to the carbon dioxide you breathed out into it. And can you tell us a little bit about the color you see?
So now it's kind of like a yellowish color, maybe a little bit green.
All right, so we're going to take those yellowish green tubes, and into one of them we're going to put a plant called Elodea. Now, Elodea can be found in aquariums. It's an invasive species, so you're actually not allowed to buy it in a lot of states. But we happen to have a bowl of it here, and I've taken a piece out. We're going to take our piece of Elodea and put it into one of these test tubes. Now, Elodea is a plant that lives, as you can see, underwater. So it's going to be perfectly content in this water. The day is getting sunny, so we're going to put these 2 tubes outside in the sun. The BTB and water by itself, and the BTB and water with the Elodea plant. You breathe a lot of CO2 into there. We're going to see if the Elodea can do anything about that. And you can cut.
Okay, stop all that. I got BTB on my face.
Ooh, their DNA's all exposed.
No, it's not. Literally, just...
Okay, so some time has passed, and we've left our Elodea out in the sun. And Lizzie, would you like to describe what you can see?
Sure thing. So the tube that just has the water in it that I breathed into is still that yellow-green color that we saw earlier. But the tube with the Elodea in it, the water is blue again.
All right, and we said that BTB turns green in the presence of CO2. So that tells us that there's less CO2 in this tube than there was at the beginning. Now there's one more cool experiment we can do. We can take this tube that has BTB, water, and the carbon dioxide you added by breathing in, and put another piece of Elodea in it, but this time, let's leave it in the dark. Okay. All right, so we have had some time for this to sit in the dark, and we can now compare our test tube that has Elodea, water, BTB, and that has been sitting in the sunlight, to the tube that had Elodea, water, BTB, and had been in the dark. All right, and what do you see, Lizzie?
So the one that was in the sunlight is still blue, probably because there's less carbon dioxide in there. But the one that was in the dark, it's still that yellow-green color.
Okay, so let's talk about data and data tables. So for this data table, it says color of BTB in tube with Elodea in light. When we started out and we put it outside, it was green. And after some time passed, you said it turned blue. The tube that we had without Elodea in the light started out green and wound up staying green. Okay. So, and just for fun, we checked to see what would happen if we didn't put the Elodea in the light. All right, so we have lots of data there. We are not going to do the oxygen saturation in here, but I think we can guess what might happen to oxygen with this plant, and if we can't, you'll find out in the reading. All right, that's it.